Hello everyone, I'm Kristen Knudsen, one of the PGY1 pharmacy practice residents at Banner University, and today I'll be talking about urine drug screening. Today's objectives are to compare the different urine drug screening modalities, describe how substances are detected by urine drug screens and what they are detecting, and recognize substances that may cause false positive and false negative results. This is a review of the two types of drug testing. First, we have immunoassays, which are a screening-only test. These are very inexpensive and rapid, and they use antibodies to detect the presence of a drug or its metabolites. The results from this test are considered presumptive until they're confirmed with gas chromatography mass spectrometry, also known as GCMS. This is the gold standard for confirmatory testing, and it has a sensitivity and specificity of 99%. It essentially breaks down the drug molecule into ionized fragments and then identifies the substances using a mass spectrometer. It actually also quantifies the amount of drug or substance present in a sample. This is just a list of the common drugs tested for in the urine. The typical 5-panel drug test includes amphetamine, cocaine, marijuana, opiates, and PCP. This table displays the federal mandated cutoff levels for the workplace developed by the Department of Health and Human Services. In some cases, as you can see, the initial test analyte differs from the confirmatory test analyte, and the confirmatory test levels are much lower than the initial test level. In testing for cannabinoids, it's important to know that the immunoassay most commonly detects 11-nor-9-carboxy delta-9 THC, which is the major inactive metabolite of THC. GCMS specifically tests only for THC-COOH, that inactive metabolite. There are two FDA-approved formulations for THC. One is dronabinol, which is a synthetic version of THC, and the second is nabilone, which is a synthetic cannabinoid similar to THC. The dronabinol will test positive on a urine drug screen, but the nabilone will not. The opioid immunoassay is screening for the presence of codeine, morphine, or 6-acetylmorphine, as highlighted in this figure. This means that there are many opioids that will not test positive on a screen. Key drugs not detected include fentanyl, methadone, oxycodone, and oxymorphone. Also, it's important to note that these drugs have overlapping metabolic pathways, so the presence of morphine in a sample could indicate morphine, codeine, or heroin use, or a combination of these. Similarly, the presence of hydromorphone could indicate hydromorphone, hydrocodone, or morphine use, and that's why it's so important to distinguish between presumptive results when reporting this information, because you really can't be sure what the substance is. There are several commonly used benzodiazepines that are just not picked up on immunoassays. This is because most immunoassays are detecting two active benzometabolites, one nordiazepam and two oxazepam. As shown in this figure, these detected metabolites are not products of clonazepam, lorazepam, or alprazolam metabolism. These are the key benzos you would need to be aware of when screening patients. This is a summary of common medications that may cause false positive results in urine drug testing. As you can see, there are a laundry list of medications that can cause false positives, so it's good to always have this resource as a reference. There are many adulterants that can be used to prevent detection of drugs. Most household adulterants, though, except for eye drops, can be detected by routine integrity measurements, including temperature, specific gravity, and pH. In conclusion, the value of a drug test really depends on the ability of a practitioner to recognize the strengths and weaknesses of that test.